Hi, everybody. Welcome to Genetic Basis of Pluripotency. We're going to start with a quick little review. I think we're all pretty familiar with the central dogma, which is DNA going from RNA to protein. So we store information in DNA, transfer it to RNA as molecular RNA, as messenger RNA, and then it goes on right to that protein. What do we mean exceptions? Only those viruses that we know of. Cells don't have any. So anything living, anything that is a cell obeys the rules. Viruses, as we know, can do a little reverse transcriptase if you're a retrovirus. So you go RNA to DNA and then DNA, RNA, protein. Nobody jumps from DNA to protein. I don't care if you're a virus or not. If you start with DNA, you gotta go through the RNA. Viruses, those sneaky little guys, can go RNA to RNA and then RNA to protein. Again, they're not living, so who cares? Just kidding, people, it's a joke. All right, gene expression. Sometimes we get it, but then it kind of blows our heads up. We have to think about every single cell has all 46 chromosomes. That means all genes in every cell. Except what? What's the exception? Gametes. We can never say always, never, everything, nothing. It's just the nature of science. So we're going to ignore those guys. Everybody else has all the genes, same genotype in every single cell in your body, right? But then how can you have a different type of cell? How can you have skin cells and various skin cells and liver cells and all different kinds of liver cells? If we all have the same exact genes, same genotype, how do we not have the same phenotype? It's because not all genes are turned on. Our genotype in our cells is expressed by what genes are expressed, not what genes we have in our cells or in our chromosomes, what genes are actually turned on in that cell type. So it's only a small subset that's turned on at any given time. And so during development, right, if you start, as we talked before, as our zygote, ooh, Mr. Zygote, he's got all the genes ready to go, right? He makes a whole big mound of himself, and then as soon as he starts to differentiate into our blastocyst, blastula, whatever you want to call it, right? We have that inner cell mass, and then the tropho, whatever, outside, right? So these guys can still become everything else. All their genes are available, but as they differentiate, different genes are turned off, okay? Regulated in a precise fashion. Makes sense, right? It has to. Usually at the level of transcription, and that's what we're going to think about here, or when we make RNA, right? Transcription is DNA goes to RNA. Every cell only turns on a specific set of genes and nothing else, right? So you can think about it, insulin is made in our pancreatic beta cells. There's a lot of other kinds of cells in our pancreas that aren't beta cells. Those don't make insulin. Our liver cells don't make insulin. Skin cells don't make insulin. Brain cells don't make insulin. That gene is there, right? The gene is there, but it's not turned on. Fascinating. I love gene regulation. How does it work? It is DNA sequences on promoter regions, right? The regulatory element, promoter sequences that regulate transcription. How does that work? Transcription factors, proteins bind DNA and make stuff happen. Sometimes these transcription factors are tissue specific, and so that helps control transcription. If you don't have a transcription factor available because it's not in the pancreatic beta cell, then it's not going to turn on a promoter for a neurotransmitter. Oh, fascinating. Usually it's a cassette of transcription factors. So several guys that work together to control gene regulation. It's almost never just one guy. Sometimes one might be more important, but it's generally a cassette or a group together. So here's a little cartoon of what we're saying. So what does this little arrow mean? What do you think this little arrow means? That means transcription is going on, right? This would be the gene of interest. And then this area, oh, right there, what do you think that is? Yeah, it's our promoter. What do you think the colored geometric shapes are? Oh, TF, transcription factors, proteins, bind DNA at promoter sites, turn on transcription. What happens when transcription's on? We get mRNA. We go then to protein, and stuff happens. You have phenotype. If it's the insulin gene, insulin's on, pancreatic beta cells making insulin. And just for example, right, transcription factors of another gene, right, those guys bind, that might turn on this gene in a different cell type. Or what if it's these two guys turning on a gene? What if only this guy binds in this scenario? We don't get transcription, you need both. So you can control by controlling the amount of transcription factor. This guy's got nobody binding, nothing's gonna happen. And 2 or 3, not good enough here. Very, very picky. Tissue specific gene expression, just for example, if we're talking about 2 different genes, right? So I don't know, we're gonna make it up. So this is like MyoD, that's the only thing I can think of. And this is collagen, I don't know. Collagen. I'm just making it up because I don't know what the heck I'm talking about. Both of these genes, right? Collagen and MyoD. Oh, MyoD, right? These are both genes in muscle cells. All the DNA, all the genes are there. Only MyoD's turned on in the muscle. Collagen's not. Well, I don't really know, but I'm going to say for our example. And I know in the skin, well, I guess maybe I don't really know. I'm making it up, people. MyoD is not on, right? We don't have any little arrow. We're not doing anything exciting. So collagen is on in the skin, MyoD is off. In muscle, MyoD is on, collagen is off. And this is just a little picture of a microarray. You can look at a whole bunch of different genes and then see by how dark, right, how dark this square is. That would be an example of a gene that's on. This would be an example of a gene that's off. So this would be like MyoD. This would be like collagen. Although I guess I did a bad example. So, okay, this works for this one over here. This one's on, right? But I guess I should have, now this guy's on over here, right? It was off in the muscle. I said this was on, so this really should be blank, right? I should have used this one. Okay, we're gonna go back because I wasn't planning ahead correctly. See how that works? Okay, my example is just about to get better. Here's on. Oh, look, that guy's off. So now it works. So on this guy, if that one is the MyoD, this is the collagen, right? MyoD off over here, collagen on over here. And then the other squares are all other genes that they looked at differentially expressed between these two, okay? Some of them aren't differentially expressed, right? Look at these guys. What about our little friends right here? What about these 2 genes? They're on in both. That's okay. We can have some things that are on in both. You can have lots of things that are on in both or in all your cells. But there has to be some genes that are different. Otherwise, it would be the same type of cell. It would have the same phenotype. Same genes on, same genes off, same phenotype. So that's our tissue-specific gene regulation, and that's what we call genetic factors controlling transcription. So genetic factors, I'll just write it over here in the corner. Genetic factors, are based on sequence of DNA and usually involves protein DNA binding or not. Okay, that would be genetics. So what is epigenetics? Epigenetics is outside of genetics. So outside of genetics, outside of this. So not sequence. Fascinating. Okay, what do we mean by that? It's chemical modifications of DNA. So not a sequence change. We can put a methyl group. So CH4 is methane, CH3 is a methyl group that we can attach to DNA, right? It's just chemistry. The attachment of these groups at specific nucleotides can cause silencing or repression of that area. So we're changing gene regulation. We didn't change the sequence, we just added a chemical modification. Same here, histone acetylation. I don't even know what an acetyl group is, but you know, we're gonna pretend like we remember chemistry and what an acetyl group is. Maybe you guys do, I don't. And so in this case, that is on a histone. Histone is on the protein. So in this case, we can modify DNA, change transcription. We can modify the histones, change transcription. It alters the chromatin environment. What do we mean by that? Well, let's take a look, shall we? All right, what is epigenetics? Well, if we don't remember, right here is our DNA, right? Yay, double-stranded DNA. But what is chromatin? It's not just naked DNA, right? Even open chromatin that's not during mitosis has some organization, is somewhat compact. This is the beads on a string, if you remember that from any class. You might have learned it, maybe 360, maybe not. Cell biology, I don't know. So it's wrapped around histone, so we can see the DNA guys, right? This guy that's going around here, and then these blobs in here, those are the histone proteins that make up nucleosomes. Look at that, you can read that. And so if we have euchromatin, that means open, that means histones, yes, are acetylated, and that keeps it open. Or heterochromatin, which is closed, that means there's no acetyl groups, and yes, methyl groups. So we're calling them ME, right? We can just go CH3. We know some chemistry here. Okay, what does that mean? Heterochromatin is inaccessible, it's restricted, it's bound up tight. So you can see how these guys are nice and tight together. These guys are nice and loose. So if it's squished up together, can't get transcription factors in there, can't make transcription work, can't turn on that gene. Has to be open to get some of those guys in there so they can get in there, so they can nucleosome remodel, so they can get to that promoter, more transcription factors, yay, transcription. So epigenetics is extremely important in gene regulation. Extremely. Oh boy. So here's a little picture of one epigenetic mechanism. So here's our little picture again, right? So RNA polymerase 2 and his friends wants to come in here and do some transcription, but if we have methyltransferases putting these methyl groups on our DNA shown in red, then RNA polymerase and his friends get denied. Oh, and this guy closes down into heterochromatin, right? So you can't do transcription here. And so here's, oh, look at that, see? See, I know my chemistry. Here he is, right there. That guy attaches to cytosines, and that causes the clumping you can think of the nucleosomes together, heterochromatin, RNA polymerase. Can't get in there. No luck for you. No transcription for you. All right, so epigenetics, super important in stem cells. That's why we're talking about it. In case you didn't get it elsewhere, it's here, and I believe there's some of it in that chapter that I posted. So if you're unfamiliar, you didn't do 360, 380, or you forgot all of it, or they didn't cover it, go back, read through that chapter, look up some more of the epigenetics. It's such nerdy fun. Epigenetics is really in charge of keeping stem cells. Oh, huge role in cancer progression. All about differentiation, and yep, you guessed it, aging too. So anytime we misregulate epigenetic, we get an aberrant development. So cancer, well, aging's normal, but not that we want it. I'm not saying anybody's aging here. I'm just saying epigenetics is really important. So why is it so important? Oh, I'm over here, sorry. Woo. Hey, sorry, here we go. Just, oh yeah, trying to make it easier to see. So the iPSCs, IPS cells, right? We haven't gotten there. This is just our preview. At one point, we knew, and we know physiologically, once you are terminally differentiated, once you are that pancreatic beta cell, that's all you can be. You can't become something else. You can't go back in time. You cannot become a precursor to a beta cell. You can't become a precursor to that precursor. You can't go back to being an embryonic stem cell, or even a cell from one of those germ layers, or one of your progenitors. You're it, physiologically. What do we mean by physiologically? In your body, in nature. Now, is this true for in the lab? Ooh, in the lab, yes. The power exists to turn those cells back. So in 2007, turn back a differentiated cell into a stem cell with the potential to become any cell type in your body. Not the placenta. We didn't go that far. Can't do that. But it was then why? Pluripotent, right? Everything except placenta in the cell. And so when we went and took a look at the epigenetics in differentiated cells, lots and lots of the genes are turned off by heterochromatin, right? So if you're a pancreatic beta cell, your insulin looks like this. Your insulin gene looks like that, looks like a stem cell gene. But a neurotransmitter gene in a pancreatic beta cell looks like that. It's packed into heterochromatin. It's not going to accidentally turn on a neurotransmitter if a rogue transcription factor comes by. It's packaged, it's shut down forever. So what do you think had to happen in order to get a differentiated cell to go back to being like a stem cell? We had to unpack, remove the epigenetics, unpack those genes, make it all, what was this stuff again? Euchromatin. That's why we're learning. That's why we have to know all these terms. All right, so it's just reflected in the cell's DNA, right? That's what I just said. Slide, arrange loosely, like that guy. Whoa, whoa. Genes are ready to spring into action at any moment. So signals might enter the cell and differentiation will begin. Any genes that are not needed, right? When we start differentiating, when we go to those 3 germ layers, if you went mesoderm, you're never ever going to become something in ectoderm or endoderm, because so you can turn off all those genes in any of the cells from ectoderm forward to its terminally differentiated cells. You're never going to need those. Those go to heterochromatin by epigenetics. All the mesoderm specific stay open. As you pass through and become more differentiated, Regular, the mesoderm guys turn off. You're on your path to be differentiated. Only your guys stay on. Everything is shutting off all the way behind you. Only genes that are required for your specialized function when you are in term diff will remain open and active. They're the only ones that are around for RNA polymerase to grab a hold of. And grab a hold he does. All right, so the story. How did we get there? So scientists noticed small numbers of genes were active only in stem cells, not in differentiated cells. These genes happen to be, I'll give you it, transcription factors. Okay, so a specific Japanese scientist, Dr. Yamanaka, wanted to see if introducing these back into differentiated cells could make them behave more like stem cells. So as soon as stem cells started differentiating in the lab, those genes turned off. In order to keep stem cells growing in tissue culture, those genes had to be on, and they had to be kept on. So maybe they could turn it back on. Started with 24 different genes, and they were able to take a differentiated cell and turn them back into what looks like an embryonic stem cell. Then they eliminated from the mixture, and they ended up with just 4 genes. These appeared to remodel the DNA and unlock. So what did they do? They took heterochrome and turned it into euchrome. And those cells became undifferentiated. They didn't know what they were, right? We opened up all that DNA. They're no longer specific to, they no longer have everything closed and just genes open for their differentiated state. And iPSCs were born. And then a flurry of publications came out. How did this work? Looking at, right, genes involved in pluripotency, genes involved in development, pluripotency factors, epigenetic regulators. How did those all work together to keep genes open in pluripotency versus guys that were closed, right, in differentiated cells? How did it work? We're not going to go through all these in detail. We will end up looking at maybe some of the classic papers and probably some new papers when we get to our point of let's see what's in the primary literature today. So for now, we're just looking at stem cell machinery, basis for understanding reprogramming. Again, here's those transcription factors that seem to be key. More publications, molecular control, right? Again, these genes here are the guys that seem to control that pluripotency. And we're not going to memorize all this stuff, but this is how they figured it out. What do these do? Yeah, they're on in ES cells. What happens if we turn off, right? If you do loss of function, that just means essentially turn it off. If you do that, it's an embryonic lethal. If you knock out or turn off OCT4 in an embryo, so we're talking like a mouse embryo, come on people, done, dead. In ES cells, or so that would be in a line, embryonic stem cell line or embryonic stem cells, right? If you knock out OCT4, they can't stay stem cells and they automatically differentiate. Ooh, to trophoectoderm. Remember that guy? So this is a really cool slide. We know so much about these transcription factors and what they do to help us put that puzzle together on how iPSCs really work. And just some more examples, more publications. These guys here, the big guys in charge of maintaining stemness. That's our new thing. We're going to stay stem cells. We're going to have stemness, right? Versus being differentiated. And then, right, here's our 3 layers. Woohoo! We love those 3 layers. And then again, just going through, really figuring out in just a couple years, like this is 2009. was discovered in 2007. It was an explosion, right? Exactly what they need. These are the guys, whoa, stop it. These are the guys here to self-renew, to stay stem cells, to keep that stemness. If you want to go to primitive endoderm, right? You got to turn on 6, keep on OCT4, turn off nanog, right? To get that. If you do this and you add SOX2, you get the mesoderm, okay? Neuroectoderm on this side, right? So we know how to, oh, the trophoectoderm, the we know how it works. So we can send these cells by adding and subtracting transcription factors and other proteins and take embryonic stem cells and turn them into whatever we want in the lab. Or take iPSCs and turn them into whatever we want in the laboratory. All right, there's still more. How is it determined, right? Oh my God, we know all this. Whoa, differentiation, endoderm, ectoderm, pluripotency, reprogramming. So what is reprogramming? Again, just to make sure we understand that reprogramming is what we call when we take an adult cell. So fibroblast cell, you can get this from a skin biopsy, right? So you can take a punch biopsy, somebody's arm, and do that, give him some transcription factors. Bingo, iPSC. So amazing. It's beautiful. Okay, sorry, I'm getting super excited here. Again, knowing what blocks it, what activates it, all of these guys here, we're showing this is the OCT4 gene. We know that these guys can activate or repress that OCT4, and OCT4 is huge in not being somewhere in the differentiated cell type. So, right, we lose pluripotency as we differentiate. If we want to go back, we got to add back that OCT4 because he turned off down here as we differentiate it. You want to go backwards, you gotta make him go backwards. And then there's just this last one that we're gonna look at today, right? And just this is the crazy interplay of all of these transcription factors. They even work on themselves, right? Which is pretty funny. So this KLF4 acts on the SOX2 gene, MYC, OCT4, and nanog, but it's also showing that it binds its own promoter. Same with SOX2. And it's the combination of transcription factors binding and remodeling. Some of these have to have some of that function or recruit the function of chromatin remodeling proteins to get those nucleosomes out, to open up all of those promoters to then activate those embryonic genes. So that's the genetic basis of pluripotency. I'm so glad, I'm so glad you were here with me. And thanks, and I will see you soon. Thanks so much. Bye.